Good morning. Thank you for coming to this session on xenotransplantation. I'm Joren Madsen. I can't see anything. I'm from MGH, and my co-moderator is Jason Gerberry from Bank of America, and we'll be joined by three esteemed colleagues of mine from Mass General who are currently in Istanbul at a big transplant meeting. Robin Pierson, Tatsuo Kawai, and Leo Riella are again all from MGH, and we're going to be talking to them about transplantation, the current state, and what the future looks like. Before we begin, I'd just like to say a few words. In 1954, history was made here in Boston when the first successful human kidney transplant was performed at the Brigham and Women's Hospital. Now, 70 years to the year later, history has been made again at Mass General, where the first successful pig kidney transplant occurred. So, transplantation is a miraculous therapy. It has saved the lives of tens of thousands of people over the years. The problem is there are not enough to go around. And the statistic is sobering. 17 Americans, not the world, 17 Americans die every day waiting for a transplant, every day. So it's a crisis. The organ donor shortage problem is not solvable with human organs. So one of the holy grails of transplantation is to use pig organs to solve the organ donor shortage. Problem is that we have evolved as human beings to fiercely reject pig tissues, pig organs, fiercely. Let me give you an example. We every day do kidney transplants from a human to a human using 3 drugs. And those drugs are very successful at keeping the kidney from rejecting for decades. Now, if we substituted the human kidney for a pig kidney, that organ, the pig kidney, would actually turn black and reject within minutes while on the operating table. It's called hyperacute rejection. That's how formidable the barrier to xenotransplantation is. And I'm going to make a confession in front of you. For the longest time, I was a skeptic. I didn't believe in xenotransplantation. And I was joined by a lot of my colleagues. We would go to meetings and we'd say things like, well, there are 3 critical principles in xenotransplantation, and we don't know any of them. And so I tell you that, but because something's changed. And what has changed is this CRISPR-Cas9 gene editing technology. It's seriously, it has revolutionized, transformed the field. And the point of this session is to convince you to become believers too. And to do that, I'm going to, again, rely on my colleagues from MGH, and we're going to ask them questions. Hopefully they can hear us. And I will begin, and then Jason will take over. So I'm going to begin with Robin Pierson, who is the world's expert in xenotransplantation writ large. And I'm going to kind of throw him a softball, somewhat rhetorical, since I just mentioned what one of the biggest advances is. But Robin, what are some of the other advances that are going to be necessary to bring xenotransplantation to the clinic?
Well, good morning, and thank you very much for the opportunity to participate in this session. Sorry that we're 7 time zones away, but we're delighted to be able to participate with the miracles of modern technology. The 4 advances I would point to, you've mentioned the gene editing, which in the decade of the 2010s progressed from TALENs and other technologies to CRISPR-Cas9, which allowed for the first time precise gene editing, making multiple gene edits to the same pig line, and putting those genes precisely where you wanted them under the control of the gene controlling mechanisms that are so important to their appropriate function. So in addition to the gene editing technology that came along in the 2010s, I turned back the clock 3 decades earlier to the 1980s, where Dolly the sheep was created, demonstrating for the first time that cloning of large mammals was possible. Then when we discovered a few tricks of molecular editing that made pigs less incompatible with humans or with the non-human primates that we test them in, we were able to produce those animals and to produce multiple copies of the same animal for testing, informative testing, to help us understand whether the gene edit that we'd introduced was in fact productive, effective at controlling the immune mechanism that it was designed to target. The third advance made over 3 or 4 decades using these progressively advanced and sophisticated gene-edited pigs was the identification that co-stimulation-based immunosuppression was key to getting a xenograft to survive. In contrast to an allograft, where, as Joren has just described, conventional immunosuppressants are really quite successful and very dependable, it turned out that unless you used a monoclonal antibody that blocked either the CD40 or the CD154 side of that particular co-stimulation pathway, the results, it was very difficult to prolong graft survival beyond a month or two. Whereas if you had the right pigs and if you avoided hypersensitized recipients, blockading particularly of the CD154 pathway was associated with prolongation of graft survival for many months and occasionally for several years. The field has progressed there with the initial CD154 antibodies had deleterious side effects on clotting cascade, but the molecules have been re-engineered by a number of companies, by Eledon, by Tonix, by a couple of other companies, to now have safe and apparently effective monoclonal antibodies to protect xenografts from cellular immune responses that occur later. The fourth major advance I'd point to is the identification of the specific infectious disease risks that cause trouble for the graft and for the recipient. Porcine CMV was associated with trouble in the first human recipient of a pig heart xenograft at the University of Maryland. That identified a hole in our screening strategy, which has since been repaired by advances in detection and avoidance of transmitting porcine CMV, which has since been demonstrated to be effective in the follow-up transplants that have been done. And pig endogenous retrovirus was initially thought to be a major barrier, but either the approach of knocking the PERV virus out in the pigs we're going to use, or if we use a pig that has not got that knockout, if PERV occurs, the antivirals developed to treat HIV are very effective against PERV. And so even if an infection were to occur, we have a strategy that we believe would be effective and safe to treat an infected patient. As a consequence, we can now progress using pigs that have genetic edits that we have developed over the past 3 or 4 decades. We can produce them in numbers through the cloning process. We have an immunosuppressive regimen that we trust, and we think we have a good handle on how to avoid introducing pig-associated infections into our human patients. That constellation of 4 major advances positions us for the pilot studies that are currently being undertaken of xenotransplantation.
Thank you, Robin. You can tell how enthusiastic he is. I'm going to ask that we try to shorten the answers. This be a lot of questions to go through. In summary, if I were to take what Robin has said and apply it to my 3-principle joke, I would say that now 3 of those critical principles are: 1, gene editing, as Robin suggested; 2, new immunosuppressive medications that are coming on the horizon; and 3, controlling viruses, CMV particularly, would be for me the 3 keys. So, Leo, we talked about hyperacute rejection, which causes the organ to turn black on the table, but there are other types of rejection. There's acute rejection, there's antibody-mediated rejection, there's chronic rejection. Where do you think we should focus now in terms of understanding mechanisms more clearly and applying therapeutics?
Thank you, Joren. Pleasure to be virtually with all of you. So exactly as Dr. Madsen mentioned, so the hyperacute rejection, the one happens immediately after transplantation, is something we can now predict through some of the testing that we do and some of the genetic manipulation that has been done with in the pig donor organ. So as we move forward with trials in humans, we will be observing long-term what's happening with the immune response and ensuring that the therapy that we're using are able to control. And there are mostly two types of rejections that we're going to be monitoring, both the cellular and the antibody-mediated rejection. in the preclinical model, so when this was tested in monkeys, a lot of the rejection response had been on the antibody-mediated route, and this seems to be well controlled with the anti-CD154 antibody, which is one of these new therapies that we include. The cellular rejection, it is something that we're always concerned. It is something that we saw in our patient that was transplanted, where we were able to treat and resolve completely their rejection. So rejection will always be the Achilles heel in transplantation. We always need immune suppression, at least in the short term. And hopefully in the future, we'll have tolerogenic strategies, so tolerance strategies where we won't need immune suppression, but we'll have to move to humans to really understand that thoroughly.
Thank you. So just to summarize, we have succeeded in blocking hyperacute rejection, but we still have to work on the other forms. Our eventual goal, though, as Leo mentioned, is to get to a point where we can do this without lifelong immunosuppression. That's called tolerance, and many labs are working on that now. So, Robin, I'm going to go back to you and ask you about the fact that we're mainly talking about kidneys, but other organs need to be transplanted, and indeed, 2 patients have received pig heart transplants. Can you tell us, are they all the same, or are there differences in the difficulty in achieving long-term xenograft survival across different organs?
What we've learned in our preclinical non-human primate models is that in contrast to pig hearts or kidneys, which with the gene edits we've briefly referred to and with the co-stimulation pathway blockade we've also mentioned, we can expect survival of weeks, months, years. When you transplant a pig liver or a lung into a non-human primate, or if you perfuse it outside of the body with human blood, the organs are much less well protected. One of the phenomena we see is that all the platelets get sucked out of the blood almost immediately after they're exposed to a pig liver or a pig lung. That doesn't happen with the heart or the kidney. It may have something to do with the fact that lungs and livers, the liver is busy scavenging the blood returning from the intestine, which may have bacteria and microorganisms in it. And one of the the liver performs a filtering function. Similarly, the lungs are filtering air that may have not only particulates, but occasionally bacteria in it. And so there are macrophages in the lung and in the liver that are designed to keep us safe from external infectious agents. Those systems of monocytes and macrophages appear to be particularly sensitive to some stimulus in the xeno reaction. And as a consequence, we are currently focused on asking whether removal of the pig monocytes and macrophages from a liver or a lung is going to prevent the damaged cells from escaping from the pig lung and making the recipient non-human primate sick. It is also possible that the platelet problem relates to some very molecular incompatibilities in the clotting system related to von Willebrand's factor. And we've done some work showing that if you humanize the von Willebrand's factor in the pig, that may be an additional gene edit that may be necessary for us to consider liver or lung xenotransplantation. So to answer your question, lungs and livers do seem to be harder and different, perhaps for the reasons I've explicated.
Yes, so it seems that there are differences in their ability to transplant both allografts, human to human, and xenografts. And it's counterintuitive, but liver, human to human, is probably the easiest transplant to achieve long-term survival, but it's one of the hardest in the xeno pig liver modality. Robin, I'm going to stick with you one last question before we go to Mr. Slayman, and that is the fact that there are 2 major companies now producing pigs for xenotransplantation, United Therapeutics and eGenesis. They have different philosophies about the gene edits they're creating. I was wondering if you can quickly just compare and contrast those philosophies between the two companies, which you're involved with both of them.
Sure. We are fortunate to be working with both companies. I would point first to the similarities in the gene edit strategies that both companies have used. Both companies have knocked out the 3 main carbohydrate targets that we understand to be recognized by preformed antibodies in most humans. And so that's consistent. Both companies have added human complement and coagulation cascade regulatory molecules, 1 or more in both, two in both categories, based on preclinical evidence that is important to protect the endothelium of a pig organ from injury initially. Both have an anti-inflammatory protein and a self-recognition molecule also added, a human self-recognition molecule to protect against monocyte and macrophage activation. So those are consistent edits that are across both platforms. Revivicor has added a growth hormone receptor knockout to limit the full adult size of their landrace strain pigs, whereas eGenesis has started with pigs that the background pig strain doesn't grow so big, it doesn't grow to commercial food production size, and a fully adult pig is more appropriate in adult size for its organs to be used in humans. So that's a difference. Revivicor has used PERV-C replication in competent pigs, whereas eGenesis has used their foundational technology to knock out all of the 60+ copies of pig endogenous retroviruses in their pig's genome. That effectively means that the pig endogenous retrovirus is off the table as a concern from a regulatory perspective, and that could be considered a significant advantage. As I mentioned earlier, from Revivicor's perspective, the use of the PERV-C replication incompetent pigs are probably safe, and if it turns out not to be, there are strategies that can be used to overcome it. I hope that answers your question.
Yes, there's controversy in the field about the number of gene edits that are beneficial, and some consider that there is a law of diminishing returns at some point. So I think the take-home message is that we're still learning which are the most important gene edits and which we don't really need to include. I'm going to switch now and talk about our patient, Mr. Slayman, who was the patient I mentioned who received the first pig kidney that was successful. I'm going to talk to Leo. Leo was the patient's physician. And maybe we can start off sauce, Leo, telling us about how you identified Mr. Slayman as a potential xenotransplant recipient. So
as Dr. Madsen mentioned early on, there's a huge organ shortage. So we have many patients in our waiting list that if they have an organ today, we would give them a transplant, because we know that the alternative treatment, dialysis, has significant effect in their body, in particular cardiovascular morbidity. So when we were looking for ideal candidates for the xenotransplantation, we were interested in patients who were candidates for a kidney transplant today, but had no living donor and had a predicted waiting time on the list of over 5 years. Not only that, these patients also should have been having a lot of trouble with dialysis. And the patient that we selected, he was having lots of issues with the dialysis access, which is the Achilles heel for dialysis. Once you lose your dialysis access, you cannot perform dialysis, and these patients will die. And he had failed all his upper extremity vascular access and lower extremity access, and had remained with one catheter that had issues every other week. So he was in a challenging situation where, unfortunately, there was no hope for him to get a human transplant anytime soon. And this is the patient that we decided to approach for the first xenotransplant.
Thank you. Dr. Tatsuo Kawai performed the actual surgery. He's a kidney transplant surgeon. Tatsuo, can you tell us about the operation? Was it similar or much different from a human kidney transplant?
It's almost the same as a human kidney transplantation, but actually, honestly speaking, pig anatomy is much easier than humans, so it's always single artery, and it should be very straightforward. But this particular patient, has a severe vasculopathy, so it's technically a bit more difficult than regular kidney transplantation.
So it wasn't the kidney so much as the patient's atherosclerosis that caused the troubles during the surgery, but it was done well. And how did you know the operation was a success, Tatsuo?
Well, when I saw the kidney pinked up immediately after the perfusion, usually transplant surgeon can tell the kidney transplant is successful or not by seeing the color of the kidneys. And usually, even in this human deceased donor kidney transplant, it sometimes takes 5 minutes or longer to see the better color of the perfusion of the kidney. But also, if we transplant pig kidney to non-human primates, it takes 5 minutes or longer to get the ideal perfusion. But this patient kidney immediately picked up and started to make urine. So that's the moment I thought it's successful.
Thank you. Leo, tell us about his course after the transplant. How did he do?
So the first week was remarkable because we held dialysis. He was making enough urine. All his electrolytes were stable, and we felt very positive. On the day 8 after transplant, he did have a rise on his creatinine, meaning, some evidence that the kidney was not working, and we did an immediate biopsy, and we found that he had a type of rejection, the cellular type of rejection that we briefly mentioned a little earlier. So we treated immediately that early rejection, and fortunately, we were able to completely reverse that. We have a repeat biopsy that was done 1 month after transplant and showed complete resolution. His electrolytes remained stable, his blood pressure remained stable, and so overall, we were so surprised and happy to see that the pig kidney was functioning exactly what we would expect from a human kidney transplant.
And tell us what happened in the end. How did he finally pass away?
So about day 51 after his transplant, we were seeing him 3 times a week. We saw him on the morning of where an event happened, and he was doing well, though he hadn't drank as much fluid the night before, and we had to do some adjustments on his supplementation of electrolytes. Nothing different than what we see very often in our human kidney transplants. And he walked from about 300 yards from the hospital to our clinic, and he was feeling okay. And in the evening, he started feeling not so well, and he became unresponsive very quickly. And so it was a sudden cardiac event that had occurred. Later, with autopsy results, we were able to identify that he most likely had what we call a ventricular dysrhythmia. So his heart went into an abnormal rhythm, and most likely as a combination of his diabetic disease, his known coronary disease, and a lot of scarring that has developed on his heart over his 8 years of dialysis prior to transplantation. So it was a combination of factors that made him predisposed to that. And positively, when we look at the kidney, the pig kidney that was transplanted, there was no evidence of rejection and nothing to point out that the kidney was related to his death. So it was completely unexpected in the course, but it really kind of shows that we have been able to control the cellular rejection, and most of the parameters that we hope that the kidney function would do, he was able to perform well.
So his death was more pre-existing cardiac disease and not so much anything that related to the pig kidney. So I'm going to stop here for the biology and the clinical side. I'm going to turn it over to Jason to talk more about the future of xenotransplantation.
Yeah, sure. So maybe I'll just toss it to you, Leo, with the next question, which is, where do we go next in terms of the field and advancing our understanding and applicability of xenotransplantation, specifically in the context of kidney? Are you planning to do more transplants? Will it be on a compassionate use basis, or is the field evolving to clinical trials? I know at least one of the sponsors right now is testing in baboons to file an IND later next year. So if you could maybe just frame where we are in terms of next steps.
Yeah, so the compassionate use is such a unique application, a regulatory pathway that we can go through the FDA even before we have a clinical trial. I think in this case, the FDA has been open to that option so that we can learn as much as we can in order to move successfully to the trial. I do feel that the big next step will be to do a small clinical trial, because we gather already so much information with a single patient, but until we test more patients, and also we select patients that are less sick, because the issue with the compassionate use is that we have to pick patients that are the sickest of the sick. so that we say this is a life-threatening condition and therefore we need to move forward with this transplant. But truly, we never do this for any drug development. And similarly with the Xeno, I think we have a much greater chance if we pick a selected group of patients they're not on that very risky category, so that we can better understand the whole course and remove some of the things that may confuse the interpretation of the results of a study like this in xenotransplantation.
Got it. So ultimately, next step will be to accrue data in patients where there's less confounding variables and ultimately interpretability, but also an ability to follow patients out. I think FDA is on record as talking about maybe 6 or 12 months as a durable sort of benefit. Maybe if you can talk about what you would see as sort of a clinical meaningful advance and how you'd measure that.
Yeah, I believe if we have a kidney transplant that lasts more than a year, that's a very important goal for us to achieve. And remember that we're comparing, in this case, a patient who gets a Xeno to one that remains on dialysis. And for some of these patients with advanced vascular disease, with diabetes, older age, their health may deteriorate, very quickly in 1 year if they remain on dialysis. So we feel that if we think as a bridge initially, not necessarily as a transplant that will last, multiple years, this would already be, a short-term success. And of course, we hope that in the future will lead to much longer survival.
Great. And can you talk just a little bit about the financial implications of this, if this is able to be a successful bridge for patients, but ultimately dialysis is going to be an inevitability for these patients?
Yeah, so, for us that do nephrology, so we're kidney physicians, dialysis expenses, so the financial burden that dialysis have in Medicare, it's tremendous. So 77% of the whole budget of Medicare goes to paying dialysis patients. So over $90,000 is spent yearly in maintaining these patients on dialysis, and the number of complications and hospitalizations that these patients have are extensive. So our competitor here is not human transplantation, but is actually dialysis. So if we're able to maintain the patient healthy, even if there's an initial higher investment in xenotransplantation, I do feel that this would be advantageous, I think, from the insurance perspective. And remember that we have 600,000 patients on dialysis in the U.S. and only, about 100,000 on the waiting list. And in part because, by patients staying long-term on dialysis, their health gets deteriorated very quickly and they're not candidates anymore. So the market of patients that would benefit from transplant are much greater. They're like 9 times greater or 10 times greater than what we see right now. And so this is, I think, where, excitement is going in our field, so that we can provide the best treatment for these patients with end-stage kidney disease that we truly believe is transplantation and not dialysis.
Great. And maybe to Robin, maybe if you can talk a little bit about what the critical path to advancing these therapeutic, or I guess xenotransplantation to market looks like. Initial clinical trials, size-wise, duration of follow-up, and does that enable potentially conditional approvals, or do you think more robust subsequent testing is going to be needed before xenotransplantation becomes a commercial reality?
My crystal ball's a little bit cloudy on this one. I think that it depends so much on what kind of results we observe in the initial small clinical trials, both of kidney and I would expect of heart. I think that we will learn fairly quickly whether the pigs we're currently working with the gene edits we've currently incorporated, and whether the immunosuppression that we have identified in our preclinical models, whether those are sufficient to reliably protect grafts for 6 months or a year. I think if you get 6-month heart survivals and 1-year kidney survivals on a reproducible, regular basis in your initial small series, that would offer optimism that the FDA would allow a large-scale IND qualifying clinical trial. IND qualifying clinical trials depending on how the FDA allows us to proceed, and that in turn would again depend on what kind of complication profile is seen in these patients, whether there's ability to go into less ill patients whose complication profile is likely to be less formidable. If we are able to justify proceeding in lower risk patients, the path to clinical approval is likely to be shorter I would estimate that at the shortest, we're looking at 3 or 4 years to IND qualification for any application, organ application, and realistically, it's likely to be longer than that. Nonetheless, that is a short ramp for many clinical drugs, and I think is motivated, my optimism is motivated in part by the pressing need and the high probability, in my estimation, that the results initially will be quite promising.
Can I just clarify one thing? When you say IND, are you referring to a registrational trial in 3 to 4 years?
Investigational new drug qualifying, the xenograft will be viewed both as a device and a drug, but so it'll follow both of those pathways simultaneously through the FDA regulatory process.
Just to continue for Robin and for Leo, I think, Leo, there were 6 separate companies involved in some way with Mr. Slayman's transplant. And they were kind enough to provide some very critical drugs to achieve the success we did achieve. Is that going to continue, or how are the companies going to interact to bring xenotransplantation to the clinic?
Yeah, the collaboration with industry and academia was critical to make it possible. From conversations that we've had with all the companies, I think they're all in terms of helping us, move forward the field of xenotransplantation. The need is so great, and I think our patients, and the advocacy that's coming from there, and foundations such as the National Kidney Foundation point out that we need to push the innovation forward. And I do feel that urgency now exists. We have a light in the end of the tunnel, and I think we will continue to learn. There will be obstacles, there will be challenges, but I do feel strongly that our field are finally getting together and working together to make this happen.
Maybe if and I want to work backwards here, just a question. What does early commercialization look like given the history of this space and how quickly folks move? Who will be the early adopters of xenotransplantation if an initial approval occurs in, say, the xenokidney realm? And how quickly do you see something like this scaling? And if anyone wants to field this one in terms of the manufacturing model and supply chain, what does that look like if anyone has a view on that side?
Well, probably initially the pig organs that will be used will be from cloned animals. Until we are sure we know what the right genotype is, it is not sensible to set up breeding herds at scale. That said, I think both Revivicor and eGenesis have mocked out and planned for success and are thinking about how to propagate herds by breeding. And that, of course, is much more efficient financially and more dependable and less expensive in many different ways. When we get to scale, presumably that will, the breeding approach will be the one that will be adopted. The immunosuppressive regimen hopefully will be well understood, refined, and my optimistic assessment is that it may be simpler and less difficult for patients, with less risks of long-term infectious complications. But again, that's a projection based on the encouraging results we've seen in non-human primates. Leo or Tatsuo, would you like to add something?
No, I think you pose a very good point. It is the number of transplants that we'll do in initial clinical trials will be in a certain speed to ensure that there's safety. And I think this is what the FDA want us to make sure that occurs. And the safety, I think, is number one. that there's no transmission of xenopathogens, so pathogens from the pig to humans. And number two, that we are not, by providing the organ, the xeno organ and the immune suppression, that we're not putting patients at a much greater risk compared to dialysis. So I do see initial slow inclusion of patients with some interval, but hopefully once that safety piece passes on, I think we can move on into bigger trials. Dr. Kawai.
Yeah, I agree with Leo and Robin. We have to be very careful for the initial cases. So we have to be very successful, and especially we have to be careful about zoonosis or something. Have to monitor very carefully.
And I think coming back to Joren's initial statement about the three principles underlying successful xeno, once we've identified them, it'll be sensible to have every capable transplant program doing these. Initially, it's essential that this work be done by teams that are familiar with the particular issues in xeno, and that this not be something that's viewed as ready for prime time until we do understand those first principles with some specificity. And we've identified a reproducible program of pig and immunosuppression and immune monitoring for infection as well as rejection that we can depend on and that can be translated effectively and safely to the general practice.
That was actually my next question, Robin. Just for background, MGH has taken xenotransplant on as almost a mini Manhattan project. We are the only institution that does xenotransplant experiments in heart, lung, liver, kidney, the whole bit. And so it kind of makes sense that we're doing it now. The question is it something that can be exported to every center? And are there enough transplanters out there if we get enough pigs to satisfy the needs of the public? So the question is, will everybody have the capability of doing it? And if so, do we have enough manpower to fill the need?
I think if you make transplantation a scheduled daytime procedure, you'll find that the technical aspects will be relatively easily mastered by the average surgeon. Tatsuo, am I off base?
I think he's stuck.
I think he may be frozen,
Yeah.
But I think it is likely to be able to be disseminated once the principles are understood.
And so to bring this to fruition, should we concentrate on the kidney or the heart? Leo, that's for you.
I believe the kidney has a major advantage compared to the heart, that we have that alternative, which is dialysis. And the number of patients that are struggling with dialysis is significantly greater than any other organ. So I do feel that kidney may be kind of driving, but there will be things that will be different. So and for sure, I think we're going to have to move things in parallel, and but the kidney will probably take the lead.
And we've written about populations of heart patients who don't have good transplant options or who have high probability of poor outcomes after transplant. And in those patients, there may be a subpopulation of patients for whom a xenotransplant trial is entirely sensible and who will help us learn how to do heart xeno safely and well.
If I could just summarize what I heard and kind of my layperson's understanding of it. So going from cloned animals to bred animals is something where you, cloned animals gives you better control, better comfort around safety. Bred animals allows you to scale. And then the other kind of technical issue that the field confronts is with what's the right immunosuppression regimen, and that's something that thought leaders like yourself will look to drive such that the broader transplant community has a more straightforward understanding of the approach to go to. Is that a fair summary?
I think so. Leo, you agree?
Yes.
Currently, I would say there's about 5 or 6 institutions in the world who are championing xenotransplantation, mainly here in the States, one in Germany, and it's a collaboration and a competition. But we believe strongly that collaboration is necessary to move the field forward. And are planning to host a meeting in the spring with all the major collaborators / competitors in the world to discuss what we learned with Mr. Slayman and what we learned with the 2 heart recipients in Maryland. What do you, Robin and Leo, what do you hope to achieve with that meeting?
Leo, do you want to start?
Sure. I believe that collaboration is the only way forward. And there's so many questions that remain to be answered. And I think if we don't put our heads together, we don't share and be transparent about our data, our concerns, and where we're moving forward, we will stay behind and we will go slow. And this is not what our patients want. Our patients finally are seeing potentially a light in the end of the tunnel, and I think we should work together to get them there safely. So I hope that we will see that transparency and that collaboration that will really advance the science here.
And I think the field of xenotransplantation has been notable over the past 30 years for the sense of the spirit of collaboration, even as we're competing with each other for grant funding and now for clinical opportunity. The folks who dedicated their lives to this, myself among them, feel very strongly that this is a field with tremendous potential that can only be realized if we work together to understand the problems based on transparency, good science, and collaboration.
And I guess maybe I'll just end the session by saying the collaboration extends not only to the other centers, but to pharma, to industry. We need collaboration from you all to make this work. And that's going to be just as critical as the other scientists and physicians who are doing this groundbreaking therapy. We have just a few minutes. I don't know if the questions are working here, but
Yeah, I'm not seeing any.
we're open to any questions, hands raised. Yeah.
Can you hear me?
Yeah, we can hear you.
Thank you all for doing this work, and really, genuinely, humanity thanks you. I don't mean that lightly. I do have a slightly, perhaps odd question, which is that 1 in 4 people on the planet is a Muslim, so practitioner of Islam. And there are certain discussions we've had in the past around xenotransplantation, particularly organs from pigs. So just wondering if you could just comment on that, generally the impact of that on these programs.
Good
By
question.
coincidence, on Sunday afternoon here in Istanbul, the IXA hosted a session which brought together religious experts from all major faith communities. We had a representative of the Shia Muslim community who gave a very nice talk. While there are some groups within the Muslim community who choose not to, who disagree, most among the Muslim community feel that violating the prescription against pig organs, eating pig organs does not apply to the transplant of organs to save a human life, because the Talmud and the Muslim religious groups and Muslim law has determined that to save a life is the highest priority, a theme that is shared by the Talmud and by the Jewish community, which similarly otherwise would prohibit use of pig organs. But in both faith communities, the use of pig organs would be supported by the majority of religious scholars and likely therefore their adherents, their religious adherents. That's a great question, though.
Anything else?
Thank you. Yeah, I think that's a sentiment shared by the vast majority of practitioners in that region too. Thank you.
Anything else?
I guess just my last question, in terms of like the next wave of data that we're going to get around xenokidneys, do you expect it to come more from compassionate use case studies, or do you think it's more the wait until we get initial clinical trial experience? And if we are pivoting to clinical trials, some of the key distinctions you'd expect in terms of the types of patients that are going to be transplanted?
Yeah. Our primary goal is to continue to advance the field. If the FDA is not open for a clinical trial anytime soon, and conversations are ongoing, then the compassionate use, will be the mode for us to keep learning and gathering further information. The biggest difference will be that in the clinical trial, we can set a number of inclusion-exclusion criteria that will not require to gather patients that are on the extreme end of their health, which, of course, is going to create confounding factors, as you mentioned. So the really hope is that the clinical trial will allow us to better assess. For example, in our patient, he had a very severe cardiac disease going to transplant, and there are things that we don't know if it was just related to the xenotransplant or purely his heart, and these are things that we will only learn if we're able to exclude some of these factors.
And I think for the heart, once we have I think the next few experiments, clinical experiments, will be extended access for individual patients. Once we have identified a regimen that is successful in 1 or 2 patients, then I think we'll be in a position for heart to design and proceed with a small-scale clinical trial, and that would then be, the result of that would then inform the design and conduct of a larger trial.
I think, for kidney transplant, different from the heart transplant, the graft failure doesn't mean the patient death. So even if a graft failed, we can put the patient back on hemodialysis, and we can do it safely. So we should, request FDA to approve for go on to the clinical trial at this point.
Great. Thank you all. Enjoy Istanbul, and thank the audience for staying and listening. I think next year we'll hear much more exciting news from the xenotransplant world. Thanks again.